Okay, let's start again. And the next topic is hierarchical clustering. So I'm going to start by explaining, give you an intuition of how hierarchical clustering works. And here's an example of a data set with 5 points and 2 variables. So each point here, or samples, for example, 1, 2, 3, 4, 5, have been characterized by measuring the expression of 2 proteins, X and Y. So if we do a scatter plot, plot X, comma Y, so the intensities, sample 1 in X, sample 1 in Y, and so on, we get this scatter plot. And then we would like to do some hierarchical clustering on this data. And so the first thing we need to do in hierarchical clustering is to calculate all pairwise distances between samples. Okay, so we have these by looking at the plot. We see that 3 and 5 are very close to each other, 2 and 4 are quite close to each other, and so on. These are the pairwise distances that we need to calculate. Once we have these, we need to use a clustering algorithm that is going to cluster to combine these samples based on their distances. So for example, here, if we look at these 5 points, these 5 samples, pair 3 and 5 are close to each other. So the first cluster that we are going to produce is a cluster that contains 3 and 5. Okay. So I'm going here to write 3 and 5. And this is the first cluster, and very often we represent this clustering as a kind of a tree or a dendrogram. And so 3 and 5 here form a first cluster. So there is now a new cluster, 3 and 5, that is positioned somewhere here. So now I look at all my pairwise distances, 2 and 4, 1 and 4, 1 and 3, 5, and so on. And the next pair of samples that are close to each other are 2 and 4. So I have my next cluster here, 2 and 4. And probably the center of this new cluster is somewhere here. So now I have 3 pairs, three samples to look at, 1, 3, 5, and 2, 4. And now the next pair to combine are 1 and 3, 5. New cluster here. So I have 1 that is now joined with 3, 5 here. And so I am now left with 2 clusters. So I combine 1, 3, 5 with 2 and 4. And now I end up starting with these 5 samples. I obtain the following dendrogram or hierarchical cluster where 3 and 5 have a certain distance separating them, and then I have 1 that joins them, and 3 and 5 and 1 are separated by a certain distance that is represented here along the branches. Same thing for 2 and 4, and then a distance from here to here, regrouping 4, 2, and 1, 3, 5. So that is an intuitive understanding of how hierarchical clustering works. Yes?
Do you always put the closest cluster on the left and then sort of work from there logically?
No. So you can switch these, you can turn these, you could turn these.
But there's no preferred method to do it that people like to see?
So what you would... so they are totally equivalent whether I write 3, 5, 1, 4, 2, or I could turn this around and have 1, 3, 5. That's exactly up to you. Now, and it doesn't matter here. It might matter if you want to try to have the kind of, one or the lower order to the left, then you might want to switch 1, 3, 5, 2, 4, or depending on any other metadata, this will have an influence. But that's kind of after your hierarchical clustering. And we'll see that this will have an effect when you produce heat maps. where then you might want to have some constraints as long as your hierarchical cluster is correct. Otherwise, they're completely equivalent. So how do we do this in R? So let's use the Mulvey dataset. Mulvey as an example. Mulvey dataset comes in Prolog data, and then I'm going to load it with Mulvey 2015 norm. So the first thing I need to do is to calculate my distance. And this can be done with the dist function in R. It will calculate the Euclidean distance. And what I need to provide as input is the kind of quantitative data. Okay, as a reminder, EXPRS will extract the quantitative data from this MSN set. Okay. But if we look at the dimensions here, the dimensions are 2,337 by 18. What I would be interested here is not to do clustering of all the proteins, but I would like to do clustering of the samples, so that kind of it's visually more useful. I mean, we could do both, but in this case I prefer to do the other one, so we'll want to transpose this. So now X is my temporary variable, contains a matrix of samples by proteins. And this is what I'm going to use as input to my distance. I'm going to create this variable D. And D now is an 18 by 18. Let's see, can I print it out? D is an 18 by 18 square matrix. Here, if I scroll to the very top, you see that provides the pairwise distances between replicate 1, 16 hours, replicate 1, 0 hours, 1, 24, 1, 0, and so on. Okay. So these are the pairwise distances that we didn't calculate explicitly here, but they were kind of intuitive by looking at the plot. We saw that 3 and 5 are close to each other. So this information we get by calculating the distance, all pairwise distances with the function dist. So the second thing we need to do is to actually calculate the hierarchical clustering, and this is something that we can do with the hclust function that takes the distance as input. So here we are. HCL, it tells us that the distance that we used was Euclidean. We use a clustering method called complete, and the number of objects, of samples at the bottom of our tree nodes is 18. And we can plot this HCL object, H clustering object, with plot. And here it is. So what do we observe? We have a clustering of the samples that match our experimental design, with an outlier or kind of an outgroup here with all the Zen samples. This is kind of the final differentiated stage. And then we have a relatively large cluster here with the early time points, 0, 16, 24, and then the kind of middle time points, 48 and 72 hours, and the replicates as always cluster together. That looks very nice, but it's not always going to be that nice. Also note the distance between the nodes here, the length of your branches. And for example, here, in this group, we have, or within each of these three kind of major clusters, we have relatively short distances, branch distances. So, as it happens, here we have a nice subgroup with 48 and 72. Here we have a nice subgroup with 16, 24, and 0. But to be honest, even if a 16 was grouped with the 24s and the 24 was grouped with the 16, that wouldn't be a problem because the distance between all these is relatively small. So there will be some noise. So even if we don't have a clustering that repeats the experimental design perfectly, that's not a problem.
Yes. When you say the distance, you're talking about the vertical line?
The length of these branches. You see the height of the tree. So here, large distance, relatively large distance, and large distance. So the kind of three groups, then early time points and late time points, they are well defined. They are separated by large distance. Whereas within here, okay, within these kind of subcluster, the distances are much smaller. So even if here we would have some scrambling of our time points, that wouldn't be a problem because the distances are small anyway. So a little bit of noise, a little bit of variability wouldn't be a problem at all. But so and we have this cluster. The next thing we want to do is or we have this tree, the standardgram. The next thing we want to do is to identify clusters. Which one do we want to group together? Something a little bit more formal than what we have done here by looking at them and circling them. And this can be done with the cut tree function. So HCl is my tree. So if I say HCl k equals 4, or k equals, let's say, 3, this will tell me if I wanted to have 3 groups, how would these 3 groups be? Well, it tells me that I would have numbers 1, 2, 3, because that's what I asked for. I want 3 clusters, and I would need to cut my tree.
Question, which library is the cut tree in?
I think it's stats.
Okay.
Yes. So this hclust and cut tree are all in stats, which comes with R. So cutting the tree means for example, in 10. So here I cut my tree at an let me maybe take 12. Cutting my tree here at 12 means that everything that has a distance smaller than 12 would be defined as a cluster. Here would be a cluster, all these would be defined as a cluster, and with 12, this is a cluster, and that is a cluster. So let me take cut tree with k equals 4. It tells me if I want to cut my tree and get 4 clusters, this is the clustering I get. All the ones would be together. This represents those with 0 and 16 and 24 hours. My second cluster are all those with a 2. Okay, 48 hours. 48 hours. All the 3s together, these are the 72 hours. 72. And then my fourth cluster would have 4 here, Zen, and then there is another Zen. So cutting the tree so that I specify the number of clusters.
Is there a remove add line? Or do you put up a line and then you want to get rid of it?
No, that is the base plotting. You can't remove. It's there. It's like, if I mess up my figure here, I need to erase it. The only thing is that you can't erase parts of your plot. You need to erase everything. So you need to rerun the lines of codes that you have. Now there is another way to cut your tree, and it's to say h to specify the height. So for example, if I cut my tree at a distance of 10, to here 10, and I will add a blue line. So here's what I'm supposed to get. Now, splitting the Zen clusters in two. See here. So now I want to cut my tree at a height of 10. And let me, I'm going to type this here. So we have record cut tree HCl H equals 10. And I'm going to sort the results so they're a little bit easier to look at. Okay, so my sample here are all those together that form cluster 1, form cluster 2, 3, and 4. Oh, sorry, I don't want 12. I want 10 here. Okay, and then the third replicate is on its own. That's in cluster 5. Yes?
Is there a way you can say what is the height I need for 12 clusters or something like that? Or
No. So what you would need to do, and that would actually be an interesting exercise, is try to cut it at many different heights, and then see which one produces.
But it's iterative by ourselves. There's no
Yes.
principled way of saying if I want 4 clusters, what is the height I need? Or
Yes.
if I want.
And the clusters are not necessarily what you would like to by looking at the plot, because when we look at the plot, we are not kind of linear. Right? We say, oh, I would like to have this plot, that plot, and so on. So
It's statistics by eye, my favorite.
yes, it is. But coming back to kind of these heights here, they are very small, so you can't rely on them. So the idea is to get a big overview and make sure that the overall pattern matches your expectations, or not. So I'm going to switch here to the material to talk a little bit about the effect. Oh, actually, I forgot to talk about the general introduction about. So the methods that we are going to see now, hierarchical clustering, seeing now, and then k-means, PCA, and so on, also called unsupervised learning methods, or as for those that were here last week, as Olga called them, class discovery, right? Where we have a data set without any annotation, and we want to discover structure in the data. So these are called unsupervised, as opposed to supervised machine learning or class prediction, where we have some sort of annotation. We know that these samples are healthy, these samples are have disease, have condition, and this other sample that I don't know, well, I'm going to compare its data and try to label it. That would be supervised machine learning, such as classification or regression, and Olga called these class prediction. And then something else that we haven't seen explicitly, which is class comparison or hypothesis testing. That would be, for example, using lemma, calculating the p-values in the ALL dataset, as we have done. Okay? But so what we're going to focus on now is this unsupervised learning, where we don't use any annotation. We are trying to discover structure in this data. And so there are kind of two parts of this. One that is clustering, which is exactly what we are doing now with the hierarchical clustering, or later on with the k-means clustering. And later on we'll see principal component analysis, what we're going to do there is doing dimensionality reduction. So if we have a data set with many, many dimensions that we can't necessarily visualize easily, we're going to use algorithms that are going to limit the number of dimensions, reduce the number of dimensions, but by trying, of course, to keep as much information in this reduced number of dimensions. And actually, unsupervised machine learning is really difficult. It's the most difficult part of all the machine learning. Classification and hypothesis testing is not as difficult. Unsupervised machine learning presents some real challenges, because there is no unique goal of clustering, or how do you define what a good clustering result is or bad clustering result? is. So yes, it is not an easy thing to do. And here is another example, coming back to the hierarchical clustering, why it is not so easy. And so what is done here is that we design a small test data set composed of 3 genes and 5 samples. Okay. So here's this very simple data set that we can look at here. Okay. So we have gene 1 in black, gene 2 in red, and gene 3 in green. Gene of protein. Okay. And so what we are going to do now here, if you recognize the code, we are going to do hierarchical clustering, but we are going to use 2 different types of distances. We are going to use a Euclidean distance. We used dist before, but here we are going to use uc, which is the same things, different functions that do exactly the same thing. Uc comes from biodist. And then instead of calculating a Euclidean distance, in the second plot we are going to calculate correlation between the gene profiles. And in so both cases, pairwise distances, pairwise Euclidean distances, pairwise correlations, we pass these distance matrices to each class and then we plot it all in one go. And this is what we get. Euclidean distance groups gene 1 and 3 together, and then 2. When we look at correlation, we group gene 1 and 2, and then 3. Which is not too much of a surprise, because when we look at 1 and 2, they actually correlate really well. It's nearly the same thing. But actually, when we look at the distance between these genes, 1 and 2, here for all the samples, are actually quite far away. How far away? That this distance plus this distance, that distance. Whereas 1 and 3 are very close to each other, except for sample 4 and 5. So the distance between black and red is bigger than the distance between black and green, but black and red correlate really well. And this explains that when we do this hierarchical clustering, we see very different results. So we need to be careful when we do clustering. What do we feed as input to this clustering? What kind of distances do we calculate? So one of the reasons why doing clustering is a difficult thing, because clustering requires some processing or some calculations, and these will have an impact on the final classification results. Something else that we might want to do is to scale our data. So in R there is a function scale. We'll do this automatically, otherwise we can calculate each value minus the row mean of all rows, divided by the standard deviation of all rows. And so here we have a scaled data set. So now we remove the kind of magnitude of our expression. We remove it. And so now we see that 1 and 2 are very, very close together. And then we have 3 here, where we see this significant difference between 1, 2, 3 here, and 1, 2, and 3 here. So that's the effect of scaling. We basically took the red one that was at the top and made all the intensities from all these genes for all the samples as similar as possible. And so here we then now repeat our hierarchical clustering using Euclidean distance without scaling and with scaling. And here we have the correlation without scaling and with scaling. We see that the scaling does not have an effect on the correlation. Distances appears the correlation. We still have 1, 2, 3, 1, 2, 3. But if we calculate the distance, scaling does have an effect. 1, 3, 2. After scaling, of course, 1, 2 become much more similar, and so they cluster together. So that shows that these tools are useful, but we need to be careful what kind of processing we do, because this will have an impact on our results. So let me come back here to my discussion with the length of the branches. Short branches, will become very sensitive to any pre-processing. And the hope, of course, is that large branches that define define well-resolved clusters will be more stable. But ideally, really, when you do something like that, you might want to, well, first of all, think carefully, what do you want to display? Are you interested in correlations or not? Have you scaled your data or not? Have you normalized your data? And then maybe you might want to try different clustering methods and to see whether some of the clusters that you see are stable. So this is the effect of distances, the effect of scaling, but of course the clustering algorithm as such also has an effect on the result we have. So here I take the ALL data set, but I only keep the transcripts here that were statistically differentially expressed. So I look at my adjusted p-values and only keep those that have adjusted p-values more than 0.05, so that I have a kind of dendrograms here that are manageable. And here it has 3 different clustering algorithms: complete, single, and average. And then, as we see here, we all see different results. So, yeah. These are useful techniques, but they come with their issues. So I have a challenge for you, and it's to reproduce this dendrogram here. And this dendrogram represents the relationship between the different organelle profiles in this spatial proteomics data set. So I'm going to show you how to get this data set. It's another data set that comes from prolog data. And the data set is called Hyperlopit. And my suggestion is to simplify the data set a little bit. Yeah. We don't really have to do it, but and so to load this simplified data set, it is available in the data directory. Or actually, if you update your data directory with the data.zip, this file will be available. But otherwise, I would suggest to simplify it a little bit by taking, let's see, p data.
Was the Hyperlopit data not there? What wasn't there yesterday?
The Hyperlopit data comes from a package, so it was not in the zip file, but I'm going to show you how to make it a little bit smaller.
Okay.
So you'll then be able to run this line of code, replicate equals 1. So we have 2 replicates in this data set, so we want to keep only maybe the first replicates. So let's call this R1. So if I say Hyperlopit, this now selects all the columns that match my first replicate. And then another suggestion would be to say marker. Oh. So this will only keep the proteins that have already an annotated subcellular localization. So by running this, we reduce the data set from a couple of 4,000 or 5,000 or so by 20 to 926 by 10. And so what you need is the expression data, proteins in samples. But instead of doing this calculation on this data set immediately, and this would give us kind of a tree with 926 leaves, final nodes, and this is the kind of tree that becomes difficult to, reason with, to visualize. Is to say, well, let's not use all the proteins, but let's use a subset of proteins. Sorry, let's see. So here, run this. Among these 900 or so proteins, we have 27 that are annotated as belonging to the 40S ribosome, 43 annotated as 60S ribosome, 13 actin skeleton, 43 cytosol, and so on. And this is available in one of the feature variables. Okay, let's extract this one to maybe something we call M. Okay, so now M is a vector that has one element per protein, and it says mitochondrion, if that protein is mitochondrial, nucleus, peroxisome, and so on. So to produce this figure, instead of producing a tree for all the proteins, my suggestion is let's calculate a mean profile for all the cytosolic proteins, a mean profile for all the proteasome proteins, and so on. And then do a hierarchical clustering on these kind of summarized results. Because this is something that is manageable, that we can look at, that we can see. If we produce a hierarchical clustering with close to 1,000 proteins, this would not be very useful. I don't remember how Stephen coined this thing, but although these are methods that we can use on large data sets, it's not going to be useful to produce visualization that we can't handle. So let's calculate mean profiles for these proteins based on where they belong and then produce a tree. Yes?
What package is that marker set?
So the data set is in Prolog data. And it's called Hyperlobby 2015. So by calling these two,
I can't get the marker inside.
oh, sorry, yes, that is, yes, you're right. That is in a package called Prolog. That function is in Prolog. So going here to kind of temporarily create this other, So I have these two variables, X that contains my matrix of interest, and M that defines the grouping in these different rows. Rows of my matrix are the proteins, and M defines the grouping. So we could probably use dplyr here, but I thought using using this by function in base R does the trick, and it works like this. It takes this matrix X, so here I have my proteins, there is my samples, and then M is here a vector that defines my groupings, and they don't need to be ordered. And so it's going to split this function, this matrix, sorry, based on the groupings defined in M, really like group by in dplyr. And then it's going to apply this function to each subset. So this first function here, it's going to, I have these columns here, it's going to calculate the colmeans. So the mean of this column, the mean of that column. And then for my second sub-matrix, going to calculate the means. So the output here would be 3 elements, 3 vectors. Oh, sorry, in this case, 1, 2, 3, 4. 4 elements that every time contain the mean profiles, the column means. So, for example, this is the mean profile for the proteasome. I thought we have the mean profile for the plasma membrane. And now I want to use the call function to R-bind these elements. Okay, to create a matrix, let me call this X again, to create the following matrix where I have my organelles of interest, and then for each organelle I have the protein profile. That is the result of calculating the column mean of all the proteins of this organ. And this is what I can now use for my hierarchical clustering. I need to choose a distance, in this case Euclidean distance, h-clust for my hierarchical clustering, and then I plot this. Yes, I don't know why. idea why it plots it the other way. Maybe this shouldn't matter, but yeah, I have no idea why it prints it upside down. Has anyone reproduced this weird behavior? Does it plot properly for you? Yeah, that works?
Yeah.
Okay.
Maybe try pulling it out and putting it back in, because one of mine, I couldn't read it, and then I just double clicked in it.
Sorry, how do you do that?
I don't know, I zoomed, and then it came out with some window. Yeah, it's better.
Ah, there you go.
And then when you, I closed it.
And then if I close it.
Right. Let's do that.
Maybe, so I suspect it's related to the fact that the names here are very long and the screen is small. But anyway, that is, yeah, that is how this could be computed. So, yes?
Would it be very difficult to do this with the pipe with dplyr? Or would you mind if you have, maybe I can put it on a sticky note, but if we could have how you do this in
Yes.
dplyr, because my first thought was I was going to start doing the pipes thing.
Yes, you could do it too. So, and that's something we could maybe do for the additional practice in the afternoon, where we would create a white data frame. Actually, there are exercises later on that will do
Okay, then that's fine.
a similar.
I don't want to derail you, that's fine.
A white data frame where we have all the samples here, and here is this one column of metadata. So this is a white data set. So we would need to gather everything except this column here into a long data set. And then we could say group by marker, group by marker and sample, and then take a mean.
And those would be pipes.
Yes, and then we could use dplyr, yes. Here I used non-dplyr syntax,
Which is dollars.
which is,
No.
but yeah, if you put it on a sticky note, I'll do it over lunch, and then we can do it together at 5. So let's look now at k-means clustering. And let me start by explaining a little bit like, no, let's first look at how to do k-means clustering. So to do k-means clustering, we use the k-means function. And the k-means function needs to take at least 2 inputs. The first one is the data that we want to cluster. So I'm going to use the same data set here. And the second piece of information that we need to tell k-means clustering is how many clusters to obtain at the end. This is a very difficult question. How many clusters to expect? And the reason I took this data set is because we expect to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 1, 2, 3, 4, 14 clusters, because we have annotated proteins as, you know, to belong to 14 clusters. And I'm also going to specify here n start equals 10, and we'll see later what this means. So here we go. Km. Km is the result of this k-means clustering. Maybe I can say summary to get a summary output. Yes, no, that's not very useful. And so the k-means clustering is going to take all the rows here, all my proteins, and assign each protein to a cluster. So let me look at the table. So basically it assigned 15 proteins to cluster 1, 44 to cluster 2, 151 to cluster 3, and so on. Okay. So what I'm going to do now to try to visualize these clusters. So this data set has, is it 10 dimensions? Yes. We have 926 rows and 10 columns. So this data set has 10 dimensions. So how could we visualize this data? It's a 10-dimensional data. How could we do to visualize it? So what we are going to do for now, and we will see in the next section how to improve on this, is to choose, I took 1, 3, 5, 9. Okay. Chose 4 out of these 10 dimensions to represent this data. And I'm going to add to this colors, col equals k-means clustering $cluster. Okay, so this is what we get. Again, not necessarily nice defaults in terms of colors. Some colors are repeated, the blue here, but this gives us an idea of these clustering results based on 4 out of, or the combination of 4 out of the 10 dimensions. Okay. So we have cluster. If we look at this and that dimension, we have cluster here, cluster here, cluster here. If we look along these 2 dimensions, we see that we have other clusters that are resolved. But this is a 10-dimensional data. How are we going to assess how well our clusters, how good our clusters are? We'll see that we can use principal component analysis to try to simplify this data set into along 2 dimensions to visualize them. So how does k-means clustering work? So I produced here this example data set. So 100 by 2-dimension x, y, and 100 points. And this here is how it goes. So we have first initial. So these are the points that we want to cluster, and the first step is random assignment. So here we want to assign 3 clusters to this data, or we want to cluster this data into 3 groups, 3 clusters. And so the first step of k-means clustering is random assignment of classes. Okay, we have red, green, and red class. They are randomly assigned. That is the first step. That's the initialization step of k-means clustering.
It's supervised. It's a semi-supervised?
No, it's unsupervised. It's unsupervised. And I have to say I want 3 clusters. Like in the example before, I said I want 14 clusters. And the next step is to calculate the average position for my 3 clusters based on this first initial random assignment. And so for this data set, it happens that my random center, for my red cluster is here, green cluster is there, black cluster is there. So that's iteration. I initialize randomly. My iteration is calculate the center of my clusters. And second step is to assign points to their nearest center. So all these points here are closest to my red center. All these points here are closest to my black center. All these points there are closest to my green center. And then I keep on these 2 iteration steps. from here on, I'll recalculate the center of my red points, somewhere here, center of my green points, somewhere here, center of my black points, somewhere here. And then I repeat my assignment. Red goes to the points that are close to red center, green goes to the points that are close to my green center, and black goes to the points that are close to my black center. And here's a little animation that shows us these iterations. Okay, at each iteration, I have a new center, and I adapt the membership of my clusters. And I repeat this until there is no change anymore, until reassigning or recalculating the center of my clusters and reassigning membership does not have any impact on my samples. Now, this has two important implications. The first one is that I need a certain number of iterations in this algorithm. If I only have one iteration, this would be my result. So there is one argument to the k-means, that is the number of iterations that need to be repeated, or the maximum number of iterations, and that's generally high enough for the algorithm to converge. But there is another parameter that influences the result, is the kind of initialization step. Okay, this is a random assignment. And of course, if I repeat my algorithm another time, I will get a different random assignment. And if I repeat it again, I will have a different random assignment. And so different random assignments means I will get here different centers and then different assignments. And so this is why it's important to make sure that the number of times we start k-means clustering is not only 1, but we set it, for example, to 10, to avoid repeating it 10 times to get a kind of what's the word I'm looking for? An agreement between these repetition of my clustering. There is another argument here that I think is called max.iter. For the maximum number of iterations the algorithm goes through, and then it would stop after, for example, 50 iterations if it hasn't converged. This doesn't happen very often, but this is an important parameter to set. And then in the notes, there is an example that shows that if we only repeat the algorithm once, we will get different results. But if we set it to 10, for example, this is going to work. Now, this is a very important argument. How many clusters to look for? And there is no magical solution for this. There are ways to get an idea of how to calculate this. And this is by using something else that is calculated as part of the k-means clustering, and that's the total within sum of squares. Here. So running the k-means algorithm on this data set with 14 clusters gives me this value for the total within squares. So what this value is that we have the 14 clusters, and we're going to calculate the within cluster distances, all within cluster distances, as sum of squares. And we are going to sum these for all our clusters. So ideally what we want are small clusters and dense clusters. And these will have relatively small within cluster distances. Okay. If we have a result with large clusters, and calculate these cluster, or within cluster, sum of square distances, and then sum them, we will end up with a total value that is bigger. So this value gives us an idea of how well our clustering algorithm performs. Okay? And this is the value that we have for 14 clusters. So what we could do, let me copy this and run it again, and I'm going to extract the total within SS. So this is the value that I get for 14 again. Let's see what happens if I calculate it for 10. What happens if I calculate it for 5? What happens if I calculate it for 30? Okay. So I can do these calculations for different numbers of clusters and look how this works and try to minimize this value to get an idea of how well different, so this is called centers, the argument here. How well we do for different values of K, or centers. And here in this code, I have done the same thing, but I have repeatedly calculated the total within squared distances for different values of K, starting with 5 to 20. And we always expect to see a curve like this. So when we start, the biggest value that we can get here would be really for a single cluster. Okay, we take all our data points and we form one cluster. And the smallest value here we can get would be if each point forms a cluster on its own. And the within total square distances, not sum of square, sorry, square distances within cluster would be 0 because each point is on its own. Okay, so the kind of theoretical extremes would be 1 and then what was it, 927. So we don't need to test all these values, we test a range of expected values. And we know that this objective value is going to decrease. That's what we expect to see. But at some point, the reduction of value becomes negligible. And this is a measure of complexity of our model. We don't want to have overly complex models because they wouldn't be any useful in this case. So what we generally do is to look for kind of an elbow, or when this decrease of total within square distances stops decreasing. It carry on. It will continue to decrease, but at some point the decrease does not become relevant anymore. So it's an empirical method, and that's one way to find a region for the number of clusters. Okay. So the one that we chose was 40, which is actually looks reasonable based on this curve, but there is no guarantee that 40 is the actual cluster based on this. It's probably around 40. So this is using the total within squared distance, and there are other similar types of statistics that can be calculated, but they always work on the same principle. We need to have a trade-off between model complexity and how well the data of the clusters are resolved. So there is a challenge here, and I suggest that maybe that would be maybe one that can be you could tackle later on for the extra practice. This challenge uses the time course data that we see here on the x-axis that are not well labeled, but the time points are first time point, second time point, third to sixth time points in every time replicates. And one thing that you could do there, and that's the result from a real paper, so I tried to use challenges that make sense. So it's a result from a real paper, where you could try maybe different values of a number of clusters with k-means, but my suggestion would be to use 16 or something around 16. So do a k-means clustering of this Mulvey 2015 norm data set to cluster the, I don't know, 2,000 proteins or so in this data set. So that is using the k-means function. But then rearrange this data set into a long table, and then use ggplot2 to produce this thing. Producing something like that with base graphics, it's not impossible, but it would be straightforward. But it's actually relatively, it's not too difficult with ggplot2. So I have a couple of steps to help you along the way here. Using k-means on this data set, use centers equals 20. I think that's a typo. It should be 16. Make sure you set and start so that your algorithm is repeated multiple times. Okay. And then you'll need to convert it to a data frame of Tibble using the expression data and then the cluster labelings that you get from running k-means. Use Gatto to transform the data in long format, and then use ggplot2 and to produce this layout, you would use a faceted. And finally, if you want to add an average profile, you would use the stat summary transformation. Or would you prefer to do this exercise now? Do you prefer to do it now? Or do you prefer to keep it for later? Let me know. What do you prefer?
What's the alternative?
Oh, we carry on with PCA, which is the next on the list. We carry on with PCA. Or you prefer to do the exercise now? What do you prefer? PCA? I have, yeah? PCA? Yes, I have 3 firm yeses for PCA, so that's what
20
we
minutes enough to do PCA.
Yeah.
Oh, but it's
Yeah. There is actually a little bit more than PCA. Right. Okay. So I'll start with PCA to show how it works. So here is a 2-dimensional data set, X, Y, that we want to analyze using principal component analysis. And so principal component analysis will look for a linear combination of these original dimensions, X and Y, so that this new dimension maximizes the variability of the data. So how can we visually estimate the variability along X and along Y in our original 2-dimensional data set? Well, the variability along X is this. The variability along Y for this data set is this. And now, the first principal component will be this dashed line here. And why is it this dashed line here? It's because it maximizes the variability of our data set. So we have that much variability along PC1, which is more than we had around X, and more than we have on Y. Okay, so we have in this case, it's a 2-dimensional data set, but this generalizes to more dimensions, 3 dimensions or more, where the PCA algorithm is going to find a new dimension that maximizes this global variability. So this is an unsupervised algorithm. It doesn't look for any annotation. It doesn't care whether this separation is related to a differential expression or any other type of annotation that we have. It looks at the global unsupervised variability to identify the first principal component, which is this new dimension, this new axis that maximizes variability. And then it will find PC2, the second axis, that has to be orthogonal to the first one. And in this case, there is only one possibility. It will be along here. And that second principal component will explain very little, actually, of variability. once this is done, we can visualize this data along these new dimensions, PC1 and PC2. But most of the variability, as you can also see from the scale here, is along PC1, and actually along PC2, we have very little variability. So this is a small example to give you an intuitive understanding how it works. And if we have two dimensions, it's of course fine to look at the two original dimensions, but if we had tens or more dimensions, this would be useful. So let's try this out. And so to do this, in R we use a prcomp function, and the prcomp takes a matrix as input. And I have used the Mulvey dataset again. And because I would like to visualize the samples again, and not the PCA of the features, I need to transpose it. And this, prcomp will produce a prcomp object that here I call PCA. So an important feature, and if I look at the summary of PCA, and I look at along I go up, along PC1 here, it tells me that the proportion of variance that is explained along the first principal component is 74. PC2 is 15%. And actually, if I look at the cumulative proportion, 74%, 90%. So this is an example where principal component analysis works really well, because along my two first principal components, which is what I'm going to use to visualize them, is that I drop information, but by just keeping the two first principal component, I still keep 90% of the overall variability that I am interested in. This is something that we can extract and calculate. So in my PCA object, I have SDEV for standard deviation. So these are the standard deviations along my 18 principal components. I have 18 initial dimensions. So I can calculate my variances. By taking the square of these. Okay, so here are my variances along my principal components, and then percentage of variance explained would be my variance divided sum of variance. Oh, this is squared. Okay. So here is the percentage of variance explained first, second, third, and so on. So I could calculate a cumulative sum . 90, 93, and so on, up to 100%. Along my 18 principal component, I explain all my variability. That's not really what I'm interested in. I want to find a certain number of dimensions that explain enough variability for me to visualize it. There is a function called biplot that can be used to visualize the result of a principal component analysis, but it is a little bit messy. So, because it overlays many things, it overlays my actual samples here in PCA. So here I have my 72 hours, the Zen, 0 hour. But here it overlays my original features that were used to calculate the principal component analysis. So biplot, I don't think is used very often. But what can be done is to extract $x. So if I look at what I have in $x is a matrix for my samples, 18 samples, and their coordinates for principal component 1, principal component 2, and 3 and 4 and so on. So I can now extract PCA $X plot to produce this PCA plot. And because I know the data set, I know that these are Zen samples, 72 hours, 48 hours, this is time point 23. Let me see what I have. So yes, this is time points 1, 2, 3, 4, 5, and 6. So what we see here is that along the first principal component is the main axis that explains the time course, where I start my early time points and I end up with my latest time point. So along this dimension, I can retrace the time of my experiment. And my second principal component allows to differentiate between the early and middle time points. So a challenge I'll ask you to do is to repeat this analysis, the principal component analysis of the multi data, and try to produce this PCA plot for time along PC1 and PC2. But could you overlay some annotation? So to do the principal component analysis, we didn't use any annotation. But if we look at $P data, so the phenotypic annotation of this data set, this is what we have. Okay. In particular, the times here. We know which samples are time points 1, 2, 3, 4, 5, 6. So this is some information that we can use, access like this, MolV 2015 $times, to annotate this PCA plot. So could you repeat this and annotate the plot, and maybe also produce the PCA plot along dimensions 3 and 4. You have all the dimensions in the X element of PCA. And let me clarify here that this is by default 1, 2, 2. But you can here use columns 3 and 4 to do the visualization along principal component 3 and 4. And could you also produce the bar plot here, bar plot, or either the kind of cumulative sum or percentage of variance explained. And this shows us, that along the first principal component we have over 90%, 70, 75%, 50%, and so on. So actually here we see that by using the first principal component, we get a good picture of what happens in this data set. So let me type this here. Repeat PCA, plot along PC1 and 2 with along PC3 and PC4. Bar plot of yes.
So in a lot of papers, I seem to call it, you have a PCA, that's not a reason, but like a PCA, and they'll say like PC1 and PC3, and there'll be a parenthesis that has a value that basically says like, what does PC, like how descriptive is PC1
Yep.
PC3? Where are those numbers coming from?
So these numbers come from what we have calculated here in the percentage of variance explained. 74% of the variance is explained by that first principal component, 50.7 by that second, 2.4 by that third. And now, later on, we'll see, I produce a lot of PCA plots for my research, and as part of the packages I develop and maintain, I have a helper function that very easily produces all these PCA plots with tons of different types of annotations I want, and that's the kind of annotation it adds to the plot. And I'll show you later on how to do it. But it's important to know where these values come from and understand how the algorithm works. And afterwards, once you find a kind of helper function that does it for you.
And PC1, because it's so high, and PC2 is so high, you might want to use, but if they co-localize on top of each other, then that would be...
So your principal components will be orthogonal of each other. So it would be very weird to get something like this, PC1, PC2, and have valid points like this. It would be weird.
But sometimes I guess what you get is you don't get like good discrimination.
Oh, I see what you mean. Yes. So
PC1 and PC2 might be all over the place, but PC1 and PC3 might actually separate.
So PC2 will always explain more variability than PC3. But you might have some resolution along PC3 that you do not have along PC2 and PC1. Yes, absolutely. So especially depending on what you have here, two principal components might not be enough. Then you will need to look along four principal components. And so principal component analysis is also an algorithm that is used to pre-process your data before running it through another algorithm. So if you have a high-dimensional data with hundreds of dimensions, and lots of these dimensions are not very informative, so what you can do is to run principal component analysis, get 100 new principal components, select the principal components that explain, I don't know, let's say 95% of your variability. Let's say these are 20 PCs, and then use this 20-dimensional data set as input to another algorithm. Yes?
How do you extract the principal components? So in PC1, 75% of the variations are explained. How do we know if that's 3 or 14 components? Can you extract that? You should be able to, right?
I'm not sure I understand your question.
So this is coming from a proteomic data set,
Yes?
right? And we're comparing different groups, and there's variation. And so I want to know what's causing that variation.
We are going to see that later after lunch, because this is not something you can get. What? No. And we'll see this in detail. What you're asking for is, what are the proteins that explain this variability?
Yes.
No, you're not allowed to do that. You will do a t-test for that. You will do a hypothesis testing analysis to tell you which are the proteins that explain the difference between two groups. We'll see this later. In other words, there is no correlation between your p-values and these things that are calculated by the principal component analysis. We'll see that. We'll see that later. Yes. Spacing has to be space or network space. I think using space is yes. 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 All of this grows after connects. Yes. Then you would have all the coordinates of protein 1, sorry, of sample 1 and sample 2 along the 18 principal components. What you want is to have coordinates for all your samples of the coordinates 1 to 4. Because when you say PCA attacks, this gives you a matrix with samples, principal components, in order. You would not get that kind of result, but if you had principal components as samples, then indeed, then you would use square bracket equation 1 column 2 column. So it is 12:30. It's official time for lunch. If you want to practice a bit more, so there's this exercise, and maybe we can do it. The results are there, and actually, I'm happy. Maybe if you have 2 minutes, I'll do it. I show the results here. So I already have my PCA object. It calculates the principal component analysis. And so to plot, to add some colors, I can use mulv dollar times. Incorrect number of dimensions, yes, because I need to extract x. So here's my PCA plot with my time points labeled. Again, it's not the nice default values, but these are the different time points. And so I could actually add a legend. With the legend function, I would like my legend to be in the top right corner. The content of my legend would be this mulv dollar times, but the unique values of this. PCH equals 1, and colors are the default ugly colors 1 to 6. Legend is missing. Let me tell it. Legend. Very ugly. I absolutely hate the bounding box around my legend. I find it the ugliest thing in the world. So, BTY box type is none. So I have to reproduce my plot and rerun the legend. Still an ugly plot, but at least it doesn't have this box. So, if I want to have a plot along PCA $x third and fourth principal component, I would do like this, and maybe adding the colors, col equals mulv dollar times, here it is. Much less resolution. And so what is we calculated the percentage of variance explained here, bar plot of this, or a bar plot of the cumulative sum PDE. And shows that I here very quickly tend towards 100% towards 1. So PC1 and PC2 do a very good job at describing this data set. Yes, sorry. So there are additional exercises that you can try out. One that maybe adds a little bit of variation. I'll change the screen here in a second. Okay. So familiarize yourself with PCA, but this time using the ALL data set. Produce a PCA plot with the samples here, and then annotate them as negative obviously or ABF. This is an annotation that we have in the field data. And then the question would be, do you think two first components would offer enough resolution? Yeah, so as opposed to this example where two first components do offer enough resolution, this would not be the case in this data set. This is maybe also an interesting one. Repeat the k-means clustering on the spatial proteomics data set. But this time, do you remember, let me show you. There was this kind of awkward visualization. And because we didn't know of PCA yet, the visualization of these 14 clusters were visualized along a 4 possible combination of original dimensions. So what you could do now is run a k-means clustering on this data set to identify the clusters, do a principal component analysis on this data set to extract PC1 and PC2, and then annotate the results of the k-means clustering, or use the results of the k-means clustering to annotate the PCA plot. And that is typically how you want to visualize any results, is using principal components. I think that's an interesting exercise. And then the last one would be to use the same data set, hyperlopit, and lopit should be in capital, 2015, and annotate this data set. Annotate it using the final assignment variable. So this is the result of the classification, saying where do the proteins belong. But then you could also use a score of this classification, or the SVM score, to transform your data. Not only using a PCA analysis to visualize this 20-dimensional data set on 2 dimensions, but annotate, color the plot based on the result of the class prediction, but use the result, the scores that we get from the class prediction, to use larger or smaller points. Use a transformation that we saw at the very beginning to add another level of resolution in your plot. The class plus the size that represents the confidence. Okay, that also combines some of the transformations that we saw before. Right, I suggest, unless you have more questions, we break for lunch and come back for 1:30. And after that, I'll briefly talk about the curse of dimensionality. We need to be very, very careful when we analyze high-dimensional data using PCA or TC algorithms. So I'll have to mention this because this is a really big problem. I'll describe the question that you had with kind of PCA versus p-values, and then we'll do a couple of exercises where you produce heat maps before we move on to the visualizing mass spectrometry data. So if you have any questions, I'll be here. Otherwise, see you just after 1:30.